Thank you, Professor Liu. As he said, my name is Cori Zuvia. I am a neuroscience major, but I do work in a toxicology lab. And today we will be looking at how passive exposure to second and third-hand cigarette smoke increases oxidative stress in children. So let's start off with some definitions. One type of passive smoke exposure is secondhand smoke. So while these individuals are not actually smoking themselves, they are receiving smoke via inhalation from the exhalations of a smoker, as well as inhalation of the smoke from the burning end of a cigarette. Another type of passive smoke exposure is third-hand smoke, which involves cigarette smoke and tobacco residues that will fall into surfaces and contaminate the environment. So, since we are working with children, this picture right here is a great example of how easy it is for children to access third-hand smoke if they live in the home of a smoker. So, these cigarette smoke residues can settle onto furniture that a child touches. These residues can settle on their clothes that the children touches, and it can also settle on their toys that they put in their mouths that they play with. And we can further see the impact of third-hand smoke on the walls of this heavy and active cigarette smoker. And it's important to note that these residues don't simply wash away. We can't wipe them off that easily. Instead, they persist in the environment for quite some time, for many years. So, considering that secondhand smoke and thirdhand smoke are not widely studied, my lab would like to dive a little bit more into this type of research. So, our project will compare levels of oxidative stress in children passively exposed to smoke versus those who have not been exposed. And we hypothesize that children passively exposed to cigarette smoke will display elevated levels of oxidative stress. So how are we going to do this? So we are working with Cincinnati Children's Hospital in a clinical research trial. They have given us 22 urine samples across 3 exposure groups. So a no exposure group, a third-hand smoke exposure group, and a mixed exposure group, which consists of children exposed to secondhand and third-hand smoke. And we have combined these last 2 groups into one passive exposure group to have more equal sample sizes here for stronger statistical analysis. And it's also important to note that within these 3 groups, we have also collected urine at a baseline time point, a 6-week time point, and a 6-month time point. So we have a lot of samples. And what we're going to do with these samples, or what we have done with these samples, I should say, is we have quantified the degree of passive smoke exposure within each group, and we have also quantified biomarkers of oxidative stress through the use of competitive ELISAs, which are enzyme-linked immunosorbent assays. So essentially, you have a 96 well plate, and the wells are going to be coated with this specific antibody. Then we're going to add in our sample, our urine sample, and it may either contain high oxidative biomarker or low oxidative biomarker. We will then also add in an enzyme conjugate, and our oxidative biomarker and enzyme conjugate will compete to bind to the specific antibody. And so if you have a lot of enzyme conjugate, more of it is going to bind to the antibody, whereas if you have a lot of oxidative biomarker, more is going to bind to the antibody. So then we add in a substrate. This substrate will interact with the enzyme conjugate to produce a colorimetric reaction. And from this color intensity, we can calculate the passive smoke exposure level or the biomarker concentration. So let's first make sure that our exposure groups are actually being exposed to smoke the way that we expect them to. So the way that we're going to do this is, the way that we have done this is by looking at cotinine, which is a nicotine metabolite, and it is correlated with passive smoke exposure. And indeed, we have found that cotinine levels are correlated with passive smoke exposure the way that we expected. So we take a look at this graph right here. We have the 3 time points that the urine was collected. And if we take a look at our passive smoke exposure group right here in purple, we can see that the cotinine levels are higher than the no exposure group, right? Indicating passive smoke exposure. We see that the no exposure group has no cotinine levels at all, which is what we would expect. And we also see here that at the 6-month time point of urine collection, the passive smoke exposure group had the highest difference in cotinine compared to the no exposure group. So now we will be analyzing all of our oxidative biomarker at this 6-month time point. So we know that our exposure groups are reliable. Now we want to make sure that our oxidative biomarker groups are reliable. So this basically means that when you collect the urine samples, there may be variables because individuals have different water intake upon urine collection. So we want to account for this by using a normalization protein. We'll be using creatinine because it is released from the body at a steady and constant rate, and it's also proportional to one's body mass, which does not change drastically daily. So we have our normalization protein, and we have found that within the two groups, the no exposure group and the passive exposure group, we see that there is no significant difference in creatinine concentrations. This is great. It tells us we can use creatinine as a normalization protein since it won't skew our oxidative biomarker anyway. It'll just make it more reliable. So now let's get to the exciting part. I keep throwing around this word oxidative biomarker. What exactly is that? What are we looking at specifically? So we are going to be looking at 8-isoprostane. This is a prostaglandin and it is formed upon fatty acid peroxidation. So essentially you have your normal fatty acid right here. This is in your body and it is going to be exposed to oxidative stress via the form of passive smoke exposure. And then it is going to be converted into 8-isoprostane right here. And upon formation of fatty 8-isoprostane, this is reducing the amount of fatty acid present in your body, right? You need fatty acids for your cells. You need it for the cell membrane. You need it for your daily bodily functions. However, 8-isoprostane, upon the formation of passive smoke exposure, is impairing the ability of your body to do its job, the ability of your fatty acids to function. And this can lead to disease such as cardiovascular disease, inflammatory disease, type 2 diabetes, and cancer. And what we have found in our data is that there is, between looking at the no exposure group and the passive smoke exposure group, there is elevated levels of 8-isoprostane concentration in the passive smoke exposure group at our 6-month time point of analysis, right? So we already saw that this passive smoke exposure group, they had elevated levels of cotinine, right? Elevated levels of exposure. Now we see that they have elevated levels of 8-isoprostane, this oxidative biomarker that is hindering the fatty acid functioning. It's also important to note that 8-isoprostane, it's highly correlated to aging and old age. Yet here we're looking at very young children. So the fact that it is present, 8-isoprostane is present at such a high concentration in the passive smoke exposure group, and it's elevated compared to the no exposure group, then we can see as of right now that these children who live in the homes of smokers are being exposed to dangerous levels of chemicals that can ultimately lead to disease. So as of right now, this study that we have collected is a pilot study. We have used it to refine our assays, and we are excited to see what happens next since we are already seeing that our passive smoke exposure group has higher levels of oxidative stress. So moving forward, we want to increase our sample sizes to strengthen our data. We may also combine these exposure groups again into one passive exposure group to have equal sample sizes. And we also want to look at additional oxidative biomarkers. So we looked at lipids, but now we want to look at DNA damage, understand lipids even more, and also look at protein oxidation and see if there's any metals in these children's urine. So we're excited to see what happens next. I just want to thank my lab for always helping me grow in research. Thank you to Mark and the NIH for funding my research, as well as Mini Grant for giving me funds for my research. Thank you to my PI, the best PI, Dr. Prue Talbot, and thank you all for letting me speak about my research today.